Hello friends, welcome back to my YouTube channel Nasrin Padasara. In this video, we are going to see 100 important topics for Icmr JRF examination section B. So first you need to know about protein structure. Along with that, you need to know about interesting facts about amino acid. So protein structure, you need to study about primary structure, secondary structure, tertiary structure and quaternary structure. Next, you need to study about Ramachandran plant. Next, structural characteristics of A form, B form, DNA, and about basic structure of DNA like purine bases and pyrimidine bases, and about Chagav's rule. Next, you need to study about type of RNA and RNA processing, like a post-translation modification, post-transcriptional modification, like that you need to study. Next, you need to give a special focus for transfer RNA. Next, you need to study about microRNA and small nuclear RNA. Next, you need to study about ribozymes, riboswitches. Next, you need to know about operon concepts like lac operon, tip operon. Next is type of enzymatic reactions and about enzymes, like list of enzymes, their cofactor and the coenzyme. Next, you need to know about enzyme kinetics, which is very important, and enzyme inhibition. You need to know about a competitive. They graphs an example of competitive, uncompetitive, and non-competitive. Next, you need to study about the plasma membrane, like lipid bilayer structure. Next, transport across the lipid bilayer, active transport, passive transport. Next, you need to know about structural organization of various intracellular organelles like cell wall, nucleus, mitochondria, Golgi, lysosome, chloroplast, like that. You need to know about the entire functions of those organelles. Next is cell division and cell cycle, like mitosis, meiosis, and also the most important thing is how the cell cycle is being regulated. Next, you need to know about cell junction, and also you need to study about extracellular matrix. Next, you need to know about cell signaling, which is very important. And next, regarding to immunological side, they will be asking antigen, antigenicity and immunogenicity, structure and function of antibody molecule. You need to study about types of antibodies like IgG, IgE, IgA. You will be getting a time table, that is differential characteristics in table in QB book. You need to know which antibody is present in plasma, which antibody can be able to cross the placenta, which antibody is present more in the blood, which antibody will be having a lesser shorter lifespan, like that you need to know. Next is antigen processing and presentation. Next is hypersensitivity reaction and autoimmune diseases. Next is vaccines. Next is immune response to bacterial diseases, parasitic diseases, viral diseases, congenital diseases, and acquired immunodeficiency. Next, you need to study about chimeric antibody, humanized antibody, human antibody, that is antibody engineering. Next, you need to study about enzymes used in recombinant DNA technology, and also you need to study about PCR and types of PCR. While studying about PCR, you need to study about thermostable polymerases. So we only know about Taq polymerase, but some other different thermostable polymerases are being identified in various locations. So you need to look forward about the thermostable polymerases that are used in PCR. Next, you need to know about analysis of DNA polymorphism, like which particular DNA polymerism technique is PCR based, which requires sequence knowledge, like that they will be asking. In addition to that, you need to study about DNA fingerprinting and DNA sequencing technology. So next, you need to study about autophagy, apoptosis, DNA replication, like enzymes involved in DNA replication, prokaryotic DNA replication and eukaryotic DNA replication, the entire process of DNA replication. Next, you need to study about DNA damage and repair mechanism and repair diseases, recombination, and next you need to know about transcription. The transcription factor, like initiation factor, elongation factor, transcription in prokaryote, transcription in eukaryotes, like that. Next is translation process, where you need to know about the structure of ribosome, process of translation in prokaryotes and eukaryotes, and you need to study about amino acylation of tRNA, translation inhibitor, and also you need to give a brief understanding about post-translation modification. In addition to that, you need to study about role of chromatin in regulating the gene expression and gene silencing. Next, you need to study about PCR type, vectors used in genetic engineering. Then in genetics, you need to concentrate on these important things like codominance, incomplete dominance, gene interaction, pleiotropy, genomic imprinting, penetrance, expressivity, phenocopy, linkage, crossing over, sex limited trait, and influence characters along with the pedigree. That's enough. Next is structural and numerical alterations of chromosome, and also you need to study about recombination, like homologous recombination, non-homologous recombination, including transposon types and site specific recombination. Okay, so the next you need to know about mutation. Next is important thing is stress physiology in plant. How plant will be coping up with their biotic and abiotic stress. Next is flowering mechanism in plant. Plant growth regulator like plant growth hormone, auxin, cytokine, and abscisic acid, like that. You need to study about all things. Next is genetically engineered for male sterility. Next you need to study about flavor saver tomato. So you need to have a general knowledge about transgenic crops and genetically modified crops. So recently mustard has been genetically modified. Currently in India they had got a license to do the cultivation of genetically modified mustard. Okay, so give a brief understanding about mustard and transgenic crops. Next you need to have a ECG and EEG, its principle, everything. Next, methods in microbiology like pure culture technique, streak plate technique. In microbiology you can refer Prescott. First 50 pages of Prescott is important. You need to study about Coach Postlet. You need to study about history of microbiology also. Next you need to have enough understanding about prokaryotic cell and eukaryotic cell. Next you need to have enough understanding about microbial growth curve and media that are employed for culturing micro. So you need to have more focus on biomedicinally important microorganisms, like which microorganism are causing disease to human and how the particular microorganism will be identified. So you need to give a special focus on healthcare related microorganism. Next, how the microorganism will be controlled, like sterilization, disinfecting techniques. Next you need to know about the DNA and RNA virus causing disease in livestock and poultry. Next, the infections in gastrointestinal tract, respiratory tract, congenital diseases, and sexually transmitted diseases. Next, you need to know about gene therapy and biological database. In bioinformatics, you need to know about biological database and the type, and you need to study about respiratory system like respiratory volume and respiratory capacity, like tidal volume, like that. Next, you need to give a special focus on high altitude physiology. What happens in high altitude? Next, you need to study about peroxisomal disorder, storage disorder like lipid storage disorder, lysosome storage disorder, and next is inborn error of amino acid metabolism. Next you need to study about mitochondrial disorder, like that. Next is you need to have a special understanding about restriction digestion, page technique, hybridoma technology. In immunology, additional to that particular thing, you need to study about TLR CD. What is present at the surface of B cell, or what is present at the surface of T cell, like that they will be asking. Next is phagocytosis, genetic drift. In evolution you need to have, a special understanding about genetic drift and founder effect. Next, in plant physiology, again you need to know about nitrogen metabolism, like denitrification, ammonification, like that you need to know. Next is C3 plant, C4 plant, scam cycle. Next you need to have a sex determination in human and also in some other things. So you can able to find a complete sex determination of all the organism in the Gilbert book. Next is staining technique in microbiology. Next is biochemical pathway, their location, their regulatory steps, the inhibitors. Next you need to know about breast milk. Okay, there were types of breast milk, you can able to check them, and types of glands and their secretion, like endocrine gland and some other gland, exocrine gland and endocrine gland. And also you need to study about more about electrophoresis with special focus about agarose and STS page technique, like that. And also you need to study about electron transport chain and the inhibitors. Last you need to study about perion diseases. So these are the 100 important topics. If you study these particular topics, means definitely you will be clearing the ICMR JRF examinations. Thank you friends, thanks for watching this video.